Hello, good afternoon to you all. The Faculty of Science of the Universidad de Valparaíso warmly welcomes you to the seminar that will be presented by Dr. Ardem Patapoutian, 2021 Nobel Prize laureate in Medicine and Physiology as part of the opening of the 2025 academic year. The cultivation of knowledge through scientific research in various areas of basic science, such as physics, chemistry, mathematics, and biology, together with the pursuit of excellence in undergraduate and postgraduate education, have shaped the development of our faculty. Furthermore, the ongoing efforts in dissemination and outreach, along with the communication and transfer of knowledge as public goods, enable us to establish a closer and more relevant relationship with global issues that have a local impact on our communities in their territories. We extend our greetings to those who are with us. Dr. Marisol Tejos, Dean of the Faculty of Science. Dr. Carolyn Weinstein, Dean of the Faculty of Pharmacy. Dr. Juan Carlos Saez, Director of the Interdisciplinary Center of Neuroscience of Valparaíso and Professor at the Faculty of Science. Dr. Ramón Latorre de la Cruz, 2002 National Prize for the Natural Science and researcher at the Interdisciplinary Center for Neuroscience of Valparaíso. Mr. Rodrigo González Redeco, Regional Ministerial Secretary of Science for the Central Macrozone. Dr. Víctor Cárdenas, Vice-Dean of the Faculty of Science. Directors of Institute and Departments, Director of Undergraduate and Postgraduate Programs. Dr. Francisco Besanilla, Professor, Department of Biochemistry and Molecular Biology from the University of Chicago. Laura Santa Cruz, Coordinator of the Science and Innovation 2030 Project. Professors and teachers, undergraduate and postgraduate students of the Faculty of Science who are joining us. We extend a special greeting to Dr. Ardem Patapoutian, 2021 Nobel Prize laureate in Medicine and Physiology. Next, Dr. Ramón Latorre de la Cruz will address you and introduce Dr. Patapoutian.
Wow, we have a full house. That's really great. Well, I am very nervous. Well, today we are joyful to have Ardem with us. Since all of you know about Ardem's amazing scientific life, an introduction to him is almost redundant. So my words will be few. Influenced by the magnificent work of Hodgkin and Huxley, who received the Nobel Prize in 1963 by their discovery of the ionic basis of the action potential, I started my life as a biophysicist having two dreams. The first was to see the electrical activity of a single channel, recalling that at that time, the mere existence of these entities was a question mark. The second one was to be able to live long enough to have the molecular structure of one of these amazing molecular machines in front of my eyes. I have been lucky enough to be present and part of the realization of these two dreams. It was Erwin Neher and Bert Sackman who, like magicians, were able to make ion channels to sing their electrical songs, opening a field that now projects itself to almost every physiological process. The development of the patch clamp technique of these two sciences was rewarded in 1991 with the Nobel Prize in Physiology or Medicines. Ion channels were everywhere. Then, it was Rod McKinnon, the first to give us the crystal structure of an ion channel, a feat many thought impossible to achieve. And because he dared to explore terra incognita, he was awarded with the Nobel Prize in Chemistry in 2003. At that time, I was dreaming about the thermodynamic principles that govern ion channels activation, and another revolution took place in the field. At the end of the last century, temperature receptors were discovered. These fantastic temperature-sensitive molecular machines are in the nerve endings of thermoreceptors and nociceptors and come in two flavors, heat and cold receptors. Ardem and David Julian were the first to clone and characterize a cold receptor, DAB, transient receptor potential melastatin 8, TRPM8 for short. And that happened to be also a menthol receptor. This study was featured as the cover story of Cell and was shortly followed by the identification and characterization of noxious coal-activated TRPA1 and heat-activated 3B3. Ardem showing that 3B3 is expressed in skin cells and not in sensory neurons. This work was highlighted as one of Science Magazine's top 10 breakthroughs of 2002. Over the next few years, Ardem Lab has demonstrated that these ion channels, TRPM8 and TRPB3, are required for thermoception. Ardem works represent a set of central insights in neuroscience. To finish, I want to mention a characteristic of Ardem's scientific life that has to do with climbing. Upon reaching the top of a mountain, most mountaineers are already thinking of the next one to climb. And the more difficult the climb, the better. Ardem could have stayed working in temperature-sensitive channels for the rest of his life. However, I think he thought like a mountaineer after the reaching of the top of the Trip Channels mountain. And he decided to climb a higher mountain. In 1987, Martinac et al. Identified the first mechanosensitive ion channels in bacteria. Mechanos transducers were also found in Drosophila melanogaster. Unfortunately, following several failed tests of candidate genes, the search of molecular bases of the sense of touch in vertebrates was not having the same success as the one enjoyed by temperature sensing. However, the situation was about to change radically. In 2010, the group of Ardem identified the receptor responsible for the sense of touch and proprioception. These two ion channels, piezo1 and piezo2, a finding that was rewarded with the 2021 Nobel Prize in Physiology. And today, we will hear the culprit himself, how these molecular machines work. Welcome, Ardem. It's a pleasure to have you here.
Thank you so much for the invitation, Ramon. To come here to see Pancho and Ramon and meet new friends has been really exciting, and a wonderful introduction for a historical perspective. Really appreciate it, and thank you all for coming. So I want to talk to you about the sense of touch today, and to introduce you, I will tell you that the sense of touch is really quite amazing. In our fingertips, we have the sensitivity to discriminate objects that are nanometer in differential. So that's really incredible if you think about it. There's people have done tests where little indentations of a few nanometers, by just touching, you know the difference. So incredible sensitivity. But beyond this quantitative measure of touch, and we all know this, that we depend on touch for our emotional health. I think this especially became very obvious during COVID years, where a hug by a loved one means so much that we were deprived of, especially in times where we couldn't do it. It became even more obvious. And many of you don't think about proprioception. It's the related sense. Instead of sensing in your skin, you're sensing how much your muscles are stretched. And from that, you know exactly where your fingers are in space. So this is how I can close my eyes and touch my nose. Or very expert musicians, shown here, Yo-Yo Ma and Emmanuel X, can play these complex tunes in their instruments without even looking at their fingers. You can see they're actually looking at each other while doing this. And we'll talk about it as well. But my favorite photo, which is kind of sad to look at right now, is this picture from the White House, where I think this illustrates how beautiful sense of touch is. So this little boy is in the Oval Office, and he tells President Obama that many of his friends tell him that his hair looks like the president's. And Barack, without skipping a beat, just bends over and says, "Touch it." And so the idea is that Barack knows that not only seeing is believing, but touching is believing. And we're not talking about this sensing the texture of the hair, but this intimate nature of affective touch, to sense that someone that has similar hair as his is in the Oval Office has very, very important meaning. So the other side of touch is pain. And although acute pain is absolutely essential for our survival, that's why we've evolved, chronic pain, which is defined as pain that persists after the initial stimulus that caused it is no longer with us, is a major unmet medical need. There's lots of statistics to prove this. Is number one reason why people go to the doctor is pain. 1 in 5 of us will suffer from chronic pain sometime in our lives. And still to this date, one of the major medications against pain is opioids, and we know that these have addictive properties, and just the overdose is a major problem in the US and I'm sure many other countries. So how do you sense all of this? Touch, proprioception, pain, all of them come from this group of peripheral sensory neurons, we call them dorsal root ganglia, that are just dorsal of the spinal cord. So this is the spinal cord. This group of neurons are very specialized, and they send processes to all your fingertips to the periphery, gather information, and send it centrally to the spinal cord. And their specializations are such that some, I've labeled them in color here, are temperature sensors and pain sensors, other are proprioceptors, other are touch sensors. And if you follow them into periphery, they innervate very different parts of your body. So for example, proprioceptors, I already mentioned, wrap around muscle, but if you zoom in the skin, there are this variety of different endings that are very specialized to sense various aspects of touch, temperature, and pain. In addition, and we'll talk about this a little bit, some of these neurons innervate your internal organs. Some of these you're conscious of, some of them you're not. But all of these are kind of little antenna in our bodies telling us what the situation is, both outside and inside of our bodies. So what we have been really interested in is how these neurons, how these cells sense temperature and pressure. So temperature and pressure, of course, are physical terms. But somehow you have to have these proteins, ion channels, that translate this energy into a chemical or electrical signal that neurons can understand. So the work of Ramon Latorre was just mentioning this, David Julius's lab, my lab, and many others, and work here done, still very exciting work on trip ion channels involved in temperature sensation that I will not talk about today. but mainly we'll focus on these piezo ion channels that are mechanosensitive, so they sense pressure. These molecules are in nerve endings at the tips of your fingertips, and this is how you sense, for example, a gentle breeze or a cactus pricking your finger. All of this started by a postdoc in the lab who came into my lab, Bertrand Coste from Marseille, that wanted to clone these mechanosensitive ion channels. And this is a 15-year-old picture when we're both much younger in front of the electrophysiology rig that he used to identify piezos. And I was talking to students today before this, and one of the questions that keep coming up is, what question do you ask and how do you answer them? And it's good to go through how we got to this. And it was actually something we did that was different in the sense that we took a question and put it in very, very simple terms of how we asked this question. So originally we're looking for these pressure sensing ion channels in sensory neurons. And although at first we were trying to find them in those neurons, we quickly realized that this is too complicated to try to answer this question. And we asked a simpler question. Since in mammals, none of the mechanically activated ion channels were identified, we asked, what if we could find a cell line, easy to work with cell that can grow in a dish, that we find a mechanically activated current there. And from there, we can clone it, identify this ion channel, and then go back to in vivo questions of where they're expressed, what are they involved in. Because other than touch, there is pain, there is hearing, there's blood pressure sensing. All of these require mechanosensitive ion channels, and none of them were known. So the way that Bertrand was looking at these is this simple cell culture dish electrophysiology, where with the glass probe, he would come and poke the cell, just like you're seeing there. And on the right, you see a recording electrode, which records the currents through the plasma membrane. And if this cell, for example, expressed the mechanically activated ion channel, you could record this as seen down there, as in the channel opens and you record the current without adding any chemicals, by just simply pushing the cell. So after Bertrand screened 30 different cell lines, he found one called Neuro2A that had these beautiful mechanically activated currents. And he said, okay, now we can knock down candidate genes one by one, record, and see when we ablate these currents. And the way he did this is used bioinformatics as well as expression analysis. He had a list of 300 genes that could be ion channels that traverse the membrane multiple times, and by RNAi technology, which was just being developed at the time, knock them out one by one and see what happens. And this is a whole year of work from Bertrand that I'm showing in one slide. What you're looking at is from Neuro2A, maximal mechanically activated currents on the x-axis of Neuro2A cells. The first is scrambled, so we're not knocking anything out. And then each of these is one candidate. So he's knocking out candidate 1, seeing how much the current is. Doesn't seem like it's reduced much. We go to second. So each of these takes about 2-3 days. So literally 11 months of negative data, day in and day out. Finally, when he knocked out candidate number 72, which was called unknown family 38 member A, he saw that there was a massive reduction in the currents. He renamed it quickly piezo 1 from Greek piezi, meaning pressure. So this is what we call, this is required, as in you knock it down, you don't see the current anymore. And we got really excited when Bertrand was able to clone the full-length cDNA, overexpress it in naive cells that are not mechanosensitive, and see massive mechanically activated currents. So you can do this with trip channels as well. So this is what a recording from this. Above what I'm showing you is how much you indent the membrane. So the first few, you're not touching the cell, and then at some point, you start pushing on the cell. And what this shows that the more you push the cell, the larger this whole cell currents that Bertrand can see. You can immediately see that it's rapidly inactivating in the sense that it doesn't stay on the whole time you push it. And this just shows control versus piezo 1 overexpressing cells. You can see massive nanoamp level currents by simply pushing the cell. So both necessary and sufficient, which biologists really love. And then this is a close family member, piezo2, which is 40% identical to piezo1, and very similar, large currents. The inactivation kinetics is slightly different than this. So we use two different kinds of recording. This is what I call wholesale recording, but you can also, by patch clamp, either in cell attach or excised patches, can inside the patch, you have a little bit of the membrane, and if you suction it, you put pressure on it, and that suction is also enough to activate the ion channel. This is actually the work of one of my alumni, Jörg Grandel, who runs his own lab at Duke, showing that you can activate this way as well, and this is much more quantitative to look at thresholds. For example, he shows that the half activation force is less than 5 millinewtons per meter. So what do these ion channels look like? Another big surprise was how large they are. So most ion channels traverse the plasma membrane anywhere from 2 to 6 transmembrane. They have 2 to 6 transmembrane domains. Piezos had 38. So that's actually more than any other protein in your genome. On top of that, structural studies that we did together with Andrew Ward, but Rod McKinnon also solved this, and by Lang Zhao, one of my trainees who went back to China, they come together as a trimer, so forming this massive, almost 1 million Dalton large ion channel. And what you're seeing here is 3 protomers coming together, each is different color. The channel actually only meets each other at the C-terminus where the pore is in the middle. The rest, which you see as ribbons, they're these transmembrane domains extending out. So one of the unusual things that you'll immediately notice, I think, is this puckered formation that they take, which is very unusual. So imagine this is the plasma membrane, and this protein is kind of as if it's almost endocytosing. And Rod McKinnon was the first one to suggest that perhaps this is the mechanism of activation. Any stretching of the membrane will flatten this channel and let ions come through. We still don't know this very well, but there has been structures in liposomes where you put them in very high stress situation. And what I'm going to show you is by Lang Zhao's work, and again, Rod McKinnon has worked on this as well, where if you put a lot of pressure on these channels, they do flatten. And again, what I'm going to show you is a morphing of 2 structures that they have solved, closed and open. So I think this is fantastic. It's, you know, an incredible molecular machine in a way that can make these massive movements that strictly mechanosensitive. And I love showing this slide, but you know, I was thinking about a year ago, what if I'm talking about this and I don't have slides with me? What if, let's say I'm in a bar talking to someone and want to talk about piezos, what do I do? So I decided to get a tattoo of this protein, and I want to show you now if you don't mind. So I thought, where can I put this tattoo so that I can show the gating mechanism, closed and open? And there was one obvious place, and it was the arm. So you can see this is close conformation. Should I come? I haven't done this in a bar yet, but we'll see. So anyways, you can imagine this is what they look like in the plasma membrane. But the problem is that all of these structures are done in artificial situations. You know, you take the protein out of the cell, you put it in detergents. And so we're getting more and more interested in, this is imagining what they look like in plasma membrane, but we want to see it in a plasma membrane. And we were thinking if we could use microscopy to look at these arms. And the idea is you lose resolution compared to cryo-EM structure, but what you gain is looking at the protein in its natural state. So can you use light microscopy to look at piezo arms? So if you're used to, you know, this is the localization precision of different microscopes, and down there is the actual size of piezo. So you can't use confocal. But over the years, these super resolution microscopes are being developed that we thought give us hope. So this is SIM, STED, STORM, and the most recent developed by Stefan Hell, this Minflux that he has a company that sells it as well, has up to 1 or 2 nanometer resolution. And we thought maybe, just maybe, we can see the dimensions of these protein in the cell plasma membrane. And so Eric Mulhall, who's very zen in my lab, and you need to be zen to do this project, took this on, and he recently published it. I'm just going to show you one slide of, but we're very excited about the future of where this could take us. And his experiments are very simple. He transfects a hex cell with piezo-1, but one that he has put artificial amino acid with an Alexaflor, very small dye, at the tips, at the ends of the arms of the piezos. He can fix the cell in different conditions and just look to see if he can see the piezo trimer. And we were blown away. I think this is probably the first ion channel intramolecular measurement of dimensions of an ion channel in the plasma membrane. And you can see he can distinguish the 3 protomers of piezo. And then once he does that, he takes an average, and these are single molecule measurements, and this is the kind of data that he gets. So on the left is AlphaFold prediction based on structure that we're getting just from in detergents. And then the next one there is normal piezo-1 expressed in hex cells. First, it's the interblade distance is longer, but look at the big heterogeneity. I'm not going to go into it, but this heterogeneity is not because we can't measure precisely. It's because there's lots of conformations. So when we look at the structure in a cryo-EM, we're averaging things. That's what cryo-EM does. So these are individual molecules. And then most excitingly, when he adds hypotonic shock to the cell, which stretches the membrane, he can see that interblade difference, the average, expands at a massive 10 nanometers. So we're seeing a 10 nanometer on average opening of the ion channel. So I don't have to get rid of the tattoo, it's real. But this is just the beginning. Eric is actually developing ways to put 7 dyes on each arm. And then so the whole idea is that in the near future, we will get more nanometer resolution of how the arms move, what is the average, if he does hundreds of molecules, what do each look like? Are the heterogeneity in the arms? So very exciting. I call it almost doing structural biology with the fluorescent nanomicroscopy. Okay, but the rest of the time I want to talk about the physiological roles. What do these piezos do in us and in animals? And we have used both loss of function and gain of function, although I will not talk about gain of function today, both in mice, other animal models, as well as from clinical data to find out what they do. So when we first looked at where piezo-2 and piezo-1 are expressed, we were very excited to see that piezo-2, the second ion channel, is very highly expressed in dorsal root ganglia, these neurons that are important for touch and proprioception and pain. And this is just the in situ hybridization in brown showing that many neurons express piezo-2. So we've made, of course, mouse models lacking piezos. And at first, there was no behavioral easy way to ask if a mouse senses touch. So we had some very primitive ideas of how to do this. And because this is the first one we've used on these knockouts, I want to show it to you. So this is what we call tape on the back. And so this is a wild-type animal. You put tape on the back, and it tries to take it off. And if you do it in a knockout, it doesn't do anything. I also show this slide to give hope to all the students to say, see what you can get Nobel Prize for. Very simple things. But we can quantitate this, and so this just shows you that once you poke the hind limb of a mouse, they lift their paw. In the wild type, the more pressure you put in, the higher percentage that they will lift it. In the knockout, you can see not at all. So they're really completely touch insensitive. How about proprioception, this sense of sensing where your limbs are in space? So without them, you'll be very coordinated. And this is another model for piezo2 knockout. And you can see these mice are very, very uncoordinated. And we don't just look at behavior. This is in vivo electrophysiology. You go to single fibers and stretch these proprioceptive nerve endings, and you can see if you increase stretch, you see this massive increase of firing of these neurons. In the knockout, 7 out of 8, we saw nothing, tiny bit in one of them, meaning that this lack of coordination is happening because proprioceptive neurons are mechanoinsensitive. So how about in humans? I gave a talk recently, and one of the ways to make people understand about proprioception is, I think, understanding, as I mentioned in the introduction, how can someone play a musical instrument without looking? If you think about it, the only way you can do this is if you have proprioception. That is, some people call it muscle memory, but what it is knowing exactly where your arms are. And one of the talks that I gave recently in Davos, Switzerland, instead of an introduction, a violinist came up blindfolded and played a musical piece, and he had never practiced to play without the blindfold. He practiced without the blindfold, but he never practiced with the blindfold till he came up. And I just want to play an excerpt of this, which I find amazing. So remarkable, all because of proprioception and all because of piezo2. And so what happens if you don't have it? Remarkably, there have been individuals that have been identified that do not have piezo2, and they completely, just like the mouse models, don't have touch sensation, don't have proprioception. So here's an individual who doesn't have piezo2, and she's asked simply to walk in a straight line with a blindfold, so that because eyes can compensate for it, she's harnessed for her own protection. And this is something that you and I can do very, very easily, but as you can see, she will have a very hard time performing this task. So how about pain? Remarkably, acute pain, so hammer hitting your finger, is almost completely normal in both animals and in the individuals without piezo2. But we also wanted to ask about chronic pain, and one of the phenotypes of chronic pain is tactile allodynia. It's two complicated words, but it's something very simple. It says when touch becomes painful. So you experience this after sunburn or after injury, where just wearing a shirt or touching becomes painful, but people suffering from neuropathic pain experience this all the time. So we showed in mice that in neuropathic pain models, this touch becomes painful is completely gone without piezo2. But because we have these individuals who have deficit in piezo2, my collaborator Alex Chesler actually tested this in humans. And the way they tested this is, imagine two boxes on your arm. One is the control, the other gets capsaicin, the active ingredient in chili pepper, and heat, which causes inflammation, and it causes tactile allodynia. Touch becomes painful. And then you can use laser Doppler imaging to show that inflammation is happening both in control individuals as well as people without piezo2. And then when you touch and ask them if it's painful or not, of course, 100% of the control individuals can tell you. The others sense nothing. So you tell them you have to guess A or B, and they guess randomly, get it right 50% of the time. So this is important for many reasons. One of them is that many targets for pain that have been identified in animal models had not translated to humans. So having some kind of verification that the same mechanism functions in humans is very important. And this and other data is making us and many others now try to find small molecules that block piezo2. We realize that it's never going to be an oral drug like you take to dock out because you don't want to lose touch and proprioception, but potentially topical, local use could be useful, or just tamping it down might be useful for patients suffering from neuropathic pain. So I talked to you about touch, proprioception, and pain. Mechanical itch is when you wear a sweater that's itchy, or a bug bites you. These are all mechanical induced itch that's also dependent on piezo. Over the years, though, we've also gotten interested in this idea of interoception, which is sensing not outside on your skin, but inside your body. And together with Steve Lieberless and Alex Chesler, we found that piezos are important for many aspects of interoception as well. Intuitively, now it makes sense after I tell you, but it's required for respiration. For each breath that you take, you monitor how much your lungs are stretched, and from this, you regulate how much you breathe. So without this, there's some disruption. Baroreception is the neuronal way of controlling blood pressure. So as blood flows on your aorta, the aorta stretches, and you sense this mechanically, and then it comes back to the autonomic nervous system to regulate your blood pressure. This is, again, piezo-dependent. This one you're very conscious of. When you have to go to the restroom, your bladder is stretching. That's what you're sensing, another form of pressure. And without piezo 2, you don't have this sensation. Indeed, these individuals that I talked to you about that don't have piezo 2 don't sense this. So when they're growing up, they get used to going to the bathroom 3-4 times a day to avoid accidents. Finally, we just had a paper a while ago showing that gut motility is dependent on piezo 2, both in animal models and in humans as well. I should mention that the condition in humans is very rare. We know less than about 50 people worldwide that have it, but more and more are coming through every day. But it's going to be rare disease no matter what. And I just want to point out that we're just starting to understand all these different senses. There's so many other places to look at. One of my favorite that Kara Marshall, one of my postdocs, is studying right now in Houston is satiety. So if you eat a large meal, the word used, I don't know what it is in Spanish, but it's full. It sounds like a very mechanical term, and we feel it as a stretch. But if you look at all the literature on nutrition sensing, when you eat, when you stop eating, etc., it's all about chemicals, proteins and fats and sugars. No one has answered this question of if you don't have the stretch sensation, will you stop eating or not? So this kind of gives us tools to look at questions that have been mainly dominated by studying more of chemical senses. So I'm not going to tell you about this, but there's a whole other literature on piezo 1. So everything I told you mainly was piezo 2, which is in neurons, but piezo 1 is in blood vessels, in bones. You might have heard that astronauts who go into outer space come back with reduced bone density. And we know now that without piezo 1, you get reduction in bone density as well. So that might be gravity sensing, the tension sensing related to it, and many stories in both red and white blood cells that I won't talk to you about. So in the rest of the time, I'm going to talk to you about a couple of unpublished work, just because why not? But before that, I just wanted to say where we're going with this. Someone could say, okay, you've studied a lot of biological functions that depend on piezo. Why don't you do something else? And the way I justify that it's important to go on is this slide. That I think we're just wrapping up stage 1. So stage 1 was kind of places where physiologists knew that mechanosensation was important, where it's baroreception or touch sensation. We said, piezo 1 does this, piezo 2 does this. But because these ion channels, you saw the structure, they're such specialized professional mechanosensors, we feel like stage 2 is to find roles of mechanosensation in areas of biology that have either been completely ignored or kind of known to have a mechano role but not studied well. The idea is that these molecules gives us a great tool to study mechanosensation where they haven't been. And the idea here is in 10 years, we'll be able to tell you that pressure sensing in your body was much more important through ion channels than we imagine now. And so one of the stories is going to be an illustration of exactly this. And this is Yung Zhao, postdoc in the lab. Just show you a few slides. Unpublished, please don't share on social media if you're taking pictures. But it's the role of mechanosensation in birthing, pregnancy. And intuitively, this makes a lot of sense, right? Uterus expands literally 500 times during pregnancy. And yet, all of the research is on hormones, etc. No one's looked at what pressure sensing does. And yet, if you go back even 50 years, there are some hints from published work that pressure sensing is important for pregnancy. For example, back in 1965, in animal models, they've put objects in the uterus that stretches it, and they see that the hypertrophy that happens in the uterus during pregnancy can happen by simply stretching it without any chemical signaling. Also, later during pregnancy, by stretching the uterus, you can see that you can get this activity of these muscles. The contractions can happen by simply stretching it. So when we encounter something like this, we say, let's take the ceiling effect, which is, let's knock out piezo 1 and 2 in every tissue related to this biology. In this case, both in the uterus and in the neurons innervating it. And we see what happens. So at first, we thought we were knocking on the wrong door, because in mice, without piezo 1 and 2, even at E18.5, which is right before birth, there was no difference whether you're losing piezo 1, 2, or both. Both total number of pups and embryo length size was completely different. But when we waited one more day, when all the pups are born, we saw a severe phenotype if you're lacking both piezo 1 and 2. So this is number of retained pups, as in pups that are not delivered. You never see it in control or individual knockouts, but many of the double knockouts had many undelivered pups. So when Yung Zhao saw this, he wanted to know what's going on during this last day, and he set up a system where he can do telemetry, so measuring contractions during that last day, and at the same time videotapes them to know when the pups are coming out. And so this is an example of the contractions of a wild-type animal, and the arrow shows when the first pup comes out, there's already contractions. When the first comes up, you see this very high level of contractions. This is for the scale night and day. And this is an example of the knockout. You see these increased contractions that are seen in wild-type doesn't happen. This was one animal. If you look at 8 control and 8 knockout, you could see that this increased contractions during the 2 hours after the first pup is born is not happening in the knockouts. And this is just actually measuring the pressure in a quantitative way to look at statistical analysis. And then from the videotaping of when the pups come, this is what it looks like for 8 different wild-type animals. Each of these thin lines is one pup being delivered. You can see in the wild-type, all of them come out around the same time at the end of day 19. And once one of them comes, the rest come out in a couple of hours. Very different story in the double knockout. You can see some of them, the first pup comes out a day and a half, 2 days later one comes, and all these orange arrows say they had incomplete labor. And again, if you quantitate this by time of interval between pups, you can see very short, less than 20 minutes in wild type, but huge variation in the knockout. And then he asked, once we see this phenotype, say why this is happening, which cells are responsible for it. And I'm not going to go through all the genetics, but he has mice that knocks out in the uterus smooth muscle cells, or in the DRGs innervating it, or in both. And the bottom line, kind of surprising new data here, is that only if you knock out piezos both in the neurons and the uterus, you see this phenotype. So I quickly went through this just to illustrate how we can find novel biology that depends on mechanosensation. And what we found is that although piezos are dispensable for pregnancy during the early stages, they have impaired these coordinated contractions the last day that's required for labor. He actually has a molecular mechanism studies, but I don't have time to talk about it. And surprisingly, this phenotype only happens if you get rid of piezo's both in smooth muscles and sensory neurons. Okay, in the last few minutes, I want to talk about another unpublished story, one of my favorite new directions, and it has something to do with wine. So that's what's cool. And I have to introduce to you the idea of chemisthesis and the mouthfeel. So chemisthesis is a bizarre word, but it actually talks about sensory experiences in your mouth that's not taste or smell. Okay, so there's many examples of this. The classic ones that we used to work on from our TRP channel work is, of course, chili peppers that activate TRPV1, the menthol, the cooling effect that affects TRPM8, and the pungency of garlic that activates TRPA1. The reason they are chemisthetic, again, is because they're chemicals, but they're giving us sensations in the mouth. Another much more ignored wing of chemisthesis involves tactile sensation. So some people call it mouthfeel. And examples are carbonated water. You know, the fizziness is not taste or smell, it's something else, it's tactile. The creaminess of yogurt or whole milk versus non-fat milk, you can immediately notice. Again, that's the texture of it. Or wine. And specifically for wine, if you like red wine, you know they have tannins, and if you're a wine aficionado, which I'm not quite there, although I'm trying to be, just tannins, these are the different words that describe various aspects of tannins. And it's quite remarkable because if I look at this list, smooth, polished, rounded, elegant, or firm, coarse, these are all tactile descriptions, right? So what do we know about wine and tannins and mouthfeel? Tannins are very complex chemicals, and they're present in grape skin, seeds, stems, as well as in the oak barrel that red wine is aged in. And one of the many properties of tannins is described as astringency, which is that drying, puckering, numbing mouthfeel. And we know this is sensed by touch neurons. It's not taste or smell. One of the proofs for this is that you can put wine on your lips and feel all of these things, which doesn't have taste or smell. So it's trigeminal, the RG equivalent of neurons. And if you read wine literature, the way people think this happens is mainly because that tannins precipitate proteins in the saliva. And by precipitating proteins in the saliva, the texture of your saliva then feels different on your tongue. That's how you feel it. That's the prevailing theory. But being molecular biologists, we asked, that's a complicated thing. Can also tannins directly affect the channel, the piezo2? And the reason this is interesting and important, what I'm going to show you is, although these channels are gorgeous and they respond to pressure, we haven't had too much pharmacology. Some of the students knew about it. We had a Yoda-1, which is, may the force be with you, kind of Star Wars piezo-1 agonist. But we screened 2 million compounds at Novartis, the pharmaceutical company, and found no piezo-2 modulators. So we put together a team of wine people in the lab and started testing something. So the first thing we did is take extracts from French oak or green tea or all of this and looked at if it has an effect on piezo2 activity. So this is not electrophysiology, this is cutting corners. This is a dye, FM143, that goes into the cell when many cation channels are open. So it's a quick way for us to monitor. And indeed, all tannin extracts reduced, presumably, the activity of piezo2. Okay, we said, then let's do some electrophysiology. And this is green tea extract. So if you express piezo2 in a cell and then put pressure on it, this is whole cell recordings. You see this current. If you do it in the presence of green tea extract, which has tannins, it's massively reduced. The same thing with French oak, you see massive reduction. But we didn't have to fractionate this because the ingredients of the major tannins in these extracts are well known. So for EGCG is the main tannic acid in green tea, and as you can see, massively blocks piezo-2. And tannic acid, the main ingredient in French oak, that's tannins, also very nicely blocks piezo-2. And this is a dose response showing that, at least for tannic acid, it's about 1 micromolar IC50. Again, this is screening 2 million compound libraries, we couldn't find it, and this is right in front of us all this time, enjoying wine, didn't think about it, blocks piezo-2. But it's not very specific to piezo-2. So tannic acid also blocks piezo-1, the sister molecule. We even went to a mechanosensitive ion channel in plants called OSCA 1.2. They have no similarity to piezos at all. It still blocks it. So it seems to block many, if not all, mechanosensitive channels, but has some specificity. It does not block our beloved temperature-activated TRP channels. Doesn't do anything to them. There's actually quite a bit of literature that tannic acid can get into the plasma membrane and affects its membrane fluidity, so that's probably how it's acting. We don't think it's a direct binding of piezos. But we've even shown that this works in the animal. So if we inject tannic acid in the paw of an animal, it reduces its touch sensitivity. It also gets rid of this tactile allodynia that I was talking about. Both of these, we know, heavily depends on piezo-2. Okay, so what I showed you is that tannins do inhibit piezo ion channels. We have a lot more data on this, but I'm just telling you instead of showing you, likely through interaction with the plasma membrane, so we don't think they're direct binders. The big question, which is not going to be easy to answer in mice, is does this piezo-2 inhibition contribute to the oral sensation of tannins in wine? And we need to do human experiments for that. If you're volunteering, let me know. What we can learn from these compounds could help us potentially be the seeds of finding molecules that could affect neuropathic pain. And I've gotten really interested in this. So, for example, there's this drink from France called chartreuse. It's an after-dinner drink that's called a digestif, and it actually has this tingling sensation in the mouth. So, for example, we have a whole project on this as well. There, it's not tannic acids, it's not tannin, so we don't know what it is. So we're fractionating these drinks and trying to see what it is. So if there's a special Chilean after-dinner drink that causes numbing or tingling, please let me know. Okay, the last 2 slides is to say that piezos are not just invertebrates. That's what we talked about most of the time, but they're present in plants, they're present in unicellular organisms, in Drosophila, in C. elegans, and there's a lot of work by us and many others. And I'll just show you one example that piezos are also in plants. And you might think, why is mechanosensation important for plants? For many, many different reasons. One of them is that as their roots grow into the soil, they have to decide if they hit something hard to go through it or to go around it. And it's thought that there's a mechanosensitive region at the tips of the roots that do this sensing and modulate the behavior of the roots. So Alireza Musavi in my lab showed that if he knocks out piezo, which is expressed exactly where the mechanosensing was supposed to be happening at the root tips, in wild type, they go right to a harder agar surface, for example. You don't see the roots very well because of how it's focused. But in the knockout of piezo in Arabidopsis, the roots cannot penetrate and go around it. You can see they're all at the surface level. It's just a fascinating example, I think, of evolution, that once something is found that can sense pressure, you can use it to sense touch, and plants use it to sense the hardness of the surface of the soil that they have to penetrate. Just wanted to thank, these are current lab members, all the alumni over the last 25 years, and key collaborators. I think I mentioned most of them. Bertrand, of course, I owe for starting this whole thing. and Yongjia was the uterus piezo stories, and the rest are in the wine group. So thank you for your attention.
We really want to thank Dr. Patapoutian for the seminar he has offered. And we are going to invite to this stage Dr. Marisol Tejos Rebolledo, who will present a gift to our guests. Yes. Finally, along with thanking everyone for participating in this activity, we extend an invitation to attend the investiture ceremony for the honoris causa doctorate awarded to the distinguished molecular biologist and 2021 Nobel Prize laureate in medicine and physiology, Ardem Patapoutian. This event will take place today at 4:30 p.m. in the auditorium of the Faculty of Social Science located at Ontaneda 2653, lo digo en español porque la mayoría de la gente sabe cómo llegar, en Valparaíso. And thank you again all, and good afternoon. Ah.
In the knockout animals, it says HOXB8. So you're only knocking out the gene in one part of the animal, and it's the hind legs, I assume, from the phenotype?
Yeah, so for each of the different, I didn't want to go through it in detail, but each of them we use different promoter crees to knock it out. The reason is because if we knock out piezo2 and or piezo1 in the whole animal, they don't survive very well. And so HOXB8, for example, is expressed from kind of lower thoracic and down. So it's one of the HOX genes, so it spares the lung breathing problem, which the mice die from. So we can then do all the tissues below the thoracic. So for the uterus, for example, that's very useful because it knocks out in all of cells, tissues in the uterus, as well as lumbar sensory neurons innervating it.
And your localization of the actual piezo in the dorsal root ganglia, that was in the cell bodies, right? But what's actually sensing the touch in Yo-Yo Ma's and Emmanuel X's fingers are here. So are these piezos also in the nerve endings?
So what I showed you was the RNA. Of course, it has to be in the cell body. But yes, we don't have good antibodies, but we and others have made fusion proteins. So for example, there's a GFP tag to the piezo2, and indeed in the proprioceptive neurons, we see it in the endings, in the part of the neurons that are going around the muscle spindles. So it's exactly where you predict that it would be. So it's at the tips of the neurons.
So they are there. Okay, thank you.
I should show that. You're right.
Thank you very much. This was really amazing. I wanted to be back to the question you said you didn't want to talk about, which is the gain of function. What happens when too much piezo in mechanosensation, at least, because you talked about many different phenotypes?
It's actually, I wish I had the time to talk about it. It's not like I don't like to talk about it. So too much piezo2 causes in humans something called arthrogryposis, which is a condition where it's a developmental disorder and the tendons are not completely extended. So people are stuck in this position. So they can't extend their... Fascinatingly, we showed that phenotype, again, this was totally not known, was because of too much piezo activity during development in proprioceptive neurons. So the whole new biology that too much activity in the neurons controls tendon development. So the details are still not completely unknown of what molecules are signaling. But yeah, so that was piezo2. In piezo1, it causes a red blood cell dehydration syndrome. And again, we recently found a role of this in macrophages because piezo1 gain of function in humans had age-onset iron overload. And we found that in mice, it's because too much activity in macrophages causes them to eat and destroy too many red blood cells, which over time causes pressure on the system and have more and more iron in the blood. Again, I like these because they're great examples of nobody had ever thought about what's the role of pressure sensing in macrophages. How would you? But lots of this information is coming from the human clinic, right? So it was because these patients had increased iron levels is why we brought it back into the mouse and tested what cell type is required, what's going on. So that's why I kind of love where we are, because it's just finding new biology and a lot of it coming from individual patients.
Thank you very much for your talk, very interesting. And I have a question about the mechanism of inactivation of the channels. And if there is some, changes in the shape of the channels, or like an active mechanism that return to the closest state of the channel, or is something else? And also, what happens if it's a stretch for a long time? You will not have sensation, no, and touch sensation.
Very little is known about this. When I got into this field, I used to, look up to people like Pancho and Ramon, and they, after we cloned the trip channels, they came in and did real biophysics and tried to understand the channel. So we need them to start working on piezo channels to answer these questions. But there is a big gap between what happens in cells and what happens in vivo, and we don't understand it. So, in HEC cells, they desensitize quickly, but in proprioceptive neurons or some other touch neurons, they fire at hundreds of hertz frequency, and we have no idea what's going on. In HEC cells, inactivation is calcium dependent, I could tell you that much, but really very little is known about it. And yeah, we need biophysicists to work on it.
Maybe 2 or 3 more questions. There in the back. Okay.
Good afternoon. Thank you for your talk. In the last part of your talk, you were talking about the tannins and its effect over piezo. I was thinking that red tea it's known to have an alleviating factor over high pressure in the blood vessels. Do you think that maybe because there's an effect of tannins in piezo2, this is a process mediated by piezo2?
That's very interesting. Has a high, don't allude to the microphone. I'm going to ask you back. What was it? Red tea affects what?
Red tea affects, alleviates the high pressure.
High blood pressure?
High blood pressure.
I didn't know that. Very interesting.
It's like what grandmas did.
Yeah.
To when they have high pressure and say, I'm going to take some tea.
Yeah. Thank you. I don't know.
Hi, thank you for the talk. I want to know if piezo 1 or 2, it's related with another ion channel, just like maybe aquaporins, because aquaporin through into the cell or out the cell molecules of water, and that is a pressure.
Structurally, piezos don't look like any other channel that we've
But
seen.
If related, if work with another ion channels.
And we haven't seen piezos work with other ion channels either. There was one study that found this, I think it's single transmembrane myodine-related protein that becomes part of the piezo and makes it actually, the inactivation kinetics becomes much slower. And that was a very nice study, not by us, but also explains how what I was talking about, that piezo in cell line could be very different in vivo, because in some situations in vivo, piezo 1 stays open as long as you stretch it. So there are going to be more and more modulators, and we talked about it with the students, cytoskeletal interactions, but direct interaction with another ion channel I haven't seen yet.
And tonings are maybe in the blood? Because you you talk about the proprioception, and maybe if the tannin are in the blood, maybe that causes another sensation. I don't know.
Yeah, I think it's the alcohol that messes up your proprioception, but don't blame tannins for it. But I don't know if the tannins survive the digestive system and are high in blood. But in wine, tannins are very high concentration, so much higher than the doses we're talking about. So.
Okay, thank you.
Thank you.
Before we finish, Ardem and Dr. Ramon, we want to invite the authorities of the government and also from the university to take a picture here with Ardem, please, if you can join us. Si las autoridades presentes de la universidad o del gobierno pueden acercarse acá para tomar una foto grupal.